So my name is Tamara Phillips. I am a professor in the Chair of the Behavioral Neuroscience Department at Oregon Health and Science University, and I'm also a Senior Research Career Scientist at the VA Medical Center in Portland.
I'd like to start by asking you how you moved along your path in alcohol research. And you go back as far as you want, if you want, to high school or something. Were there particular people that had an impact on your progression to become a scientist, become an alcohol researcher?
Well, I'm going to start a bit early on. I was moved around a lot during my childhood. My mother married 4 times before I was 15 years old. And so we moved from place to place depending upon the situation. I believe that created a lot of internal motivation for me to be very self-sufficient and independent. And I have had that characteristic. It seems that adversity can be very negative, but it also can have some positive impacts. And so it really made me a lot stronger in some ways and made me want to accomplish anything I put my mind to. And I had a lot of motivation to do that for some reason. I don't really know why, but I did. And initially, the thing I wanted to accomplish was music. And so I play the flute. I still play the flute. And I was a music major at first. I ended up being valedictorian of my 50-student graduating class from Tombstone High School in Arizona, of all places. And because I was valedictorian, I was offered a full ride scholarship to Arizona State University as a music major. And I decided to accept that scholarship. But somewhere through that first year, I started to think about how I was going to support myself and whether my track as a musician could ever lead to me being actually a professional musician. I knew I didn't really want to teach, so I started thinking about the courses I had taken that year and what interested me. And I had taken an introductory psychology course and had become really interested in the information about mental health disorders, found that to be absolutely fascinating. So at that point, I decided to make some changes. And so I made 2 decisions that I think really put me on the path to where I am now. One was to move to the East Coast, to New Jersey, where my father lived and some other family members, so that I could get to know them better. And the second was to change my major to experimental psychology. And so I joined William Patterson State University in Patterson, New Jersey, and I ended up in an honors biological psychology program, which would now be called behavioral neuroscience, and had the opportunity to take a course called behavioral genetics, which was extremely unusual at the time. Very hard to find a course like that. And I was soon thereafter asked to join as an undergraduate researcher the lab of a researcher there by the name of Martin Hahn. He went by Marty. And he used selected lines in his research, lines that had been bred for differences in brain weight, and was studying cognitive factors to see if that had any impact. And so that was my very first exposure to research. I really knew nothing about getting a Ph.D., what that meant, what it meant to be the head of a lab, or anything like that. And in fact, I was taking biology and chemistry courses outside of my psychology degree because I thought that I was going to be applying to medical school. Marty started telling me that he thought I had what it took to be a scientist. I was very, very stubborn, though, and I really didn't want to change my plans. And so I kept refusing to consider that. He asked me at one point if I would go up to the State University of New York in Albany and meet a researcher that he knew there that was a behavioral geneticist and potentially see if I might be interested in changing my mind. And I just refused. I wouldn't go. And so he brought Bruce down. He got Bruce to drive the 2 hours down to see me. And they showed up at the pharmacy where I was filling prescriptions to pay for my tuition. And they took me to lunch. And long story short, I ended up going up to SUNY Albany and seeing Bruce's lab and decided to join that laboratory. So I think that, the story about that is how people kind of push you down paths sometimes where they see some promise in you, and you take those paths. And I don't regret that. I think I have been happier being as a scientist than I may have been had I gone down the physician route. So that's sort of the first time that I was really impacted by someone who wanted to do something that they felt was best for me. So I ended up working in Bruce's lab with, again, selected lines. They were called the long sleep and short sleep lines. They were actually bred for sensitivity to the sedative hypnotic effects of alcohol. And so that's the first time that I was exposed to doing addiction research, and it was in the alcohol field. I was very familiar with the concept of selective breeding because I had worked with lines in Marty's lab, and the focus of my research ended up being on sensitivity to the stimulant effects of alcohol as a model of the euphoria that people sometimes experience after they consume low doses of alcohol. And that ended up being a major theme of my research for many years after I left and after I received my Ph.D, studying stimulation and sensitization, and maybe somewhat unique in the field.
Mm-hmm. So this is a little bit of a detailed question, but I'm dying to know: did Marty Hahn's work? Did he find a relationship between brain weight and anything?
So in short, no.
Okay.
Just as you might expect, it's not that simple.
Yeah.
They went on to do some studies, though, just looking at T-maze, cognition, et cetera, and were able to find some genetic factors that were important, but it wasn't brain weight.
Okay. But it's certainly, when no one knows, you got to look.
Yeah, that's right. That's right.
And I keep seeing themes in these conversations of people helping each other, and you get a mentor. Took a lot of insight from him to guide you.
Yes, indeed, and really the fact that he wouldn't just take no for an answer. I think that's something impressive as well. He could have just let me go off and do what I wanted to do.
Mm-hmm. What challenges have you encountered along your path?
Well, I'm not young. I was in graduate school between the years of 1981 and 1985. And the PI career at that time was really male dominated. There was not a single laboratory in my department at SUNY Albany that was headed by a woman. Every single lab was headed by a man. And all of my courses were also taught by men at that time. So my thesis committee was comprised of 3 men named Bruce, which is like a Monty Python skit.
There's a Bruce skit. Yes.
There's a Bruce skit. I know, isn't that hilarious? During that time, I did suffer some sexual harassment at a time when it wasn't spoken of, and it certainly wasn't reported. I think probably a lot of women of my era went through that. And then also I would say some disrespect. I remember when I went to the biology department to ask if I could enroll in a molecular genetics course that I wanted to take. And because I was from the psychology department, I had to get permission. And I'll never forget the instructor saying to me, well, you can enroll, but we know that it will be very hard for you, and you probably won't do that well. And, maybe we might have to, curve the grades for you. And that's all it took to motivate me to to ace the course. So sometimes challenges are put in your way, and you're up for them.
Can I ask did you ever get to see the reaction of the professor when you aced the course?
Yes. At the end, I went up and spoke with him afterwards and said, do you remember what you said to me when I said I wanted to enroll in your course? And we had a pretty nice conversation about that. And he apologized to me. So
good.
Yeah.
You had another positive impact.
That's right. Yeah. Yeah. So I thought that was a good thing too. When I reached my 4th year of graduate school, I was told that there was no longer going to be any stipend support for me. And it's because my mentor really wasn't writing NIH grants. I'm not really sure how, but he was getting his laboratory supported by the university at that time. And so my only option was to apply for these teaching fellowships that they had for people in my situation. There were only 2 of them. And so it was risky. I didn't know if I was going to get it or not. I did end up getting the fellowship, but it meant that I had to teach a a 60-student statistics course, and a 150-student intro to psych course each of the 2 semesters during my 4th year while I was trying to finish my degree and while I was continuing to take care of the animals in Bruce's laboratory. So basically, I was working for free in his laboratory by being, while being paid for teaching.
Did you have to create all those lectures new?
Yes. Yes. It was an astronomical amount of work. I think back now, I wonder how I got through this period of time. So that delayed my degree by 6 months. I got finished in 4.5 years still, which is pretty quick.
I know that's less than the average when I was in grad school.
Yeah. Yes. And that last 6 months, I didn't have a job of any kind. So I was using money that I had managed to save or that my family could give me to support myself during that last 6-month period. So that was a bit of a tough time, a hurdle to say the least. After that, I accepted a postdoctoral position and was able to move on. So I still feel that although I had those kinds of hurdles in front of me, I still managed to get through, and really with no hard feelings between Bruce and I. Really, I didn't feel it was his fault. So
yeah. I don't know if you feel this way, but there are times when I have felt like I can't do it, but I get it done, and then I realize, oh, I can do that.
yeah,
And the lesson is that we have more capacity often than we think, and then
I agree, and I think that's one of those things that I've had throughout my childhood, is that motivation to succeed no matter what. And even then, and you know what, I taught that statistics course, and most people shy away from teaching statistics. I was lucky because Bruce was the graduate level statistics instructor at the university, and so I was very well trained. And I know that I impacted some of the people in that statistics course who thought they couldn't do it because I was so motivating, telling them that yes, you can do this, and saw them through. So in some ways, again, that was really a very positive experience. Not to mention that I now had a lot of teaching under my belt, which put me in good position for a postdoc to be allowed to do more teaching. So I wasn't a TA, I was actually the instructor, which is pretty unusual at that level.
it is.
Yeah.
What brought you to OHSU?
So my first postdoc was at the Institute for Animal Behavior at Rutgers University, but it was the Rutgers campus in Newark, New Jersey, not the nice one.
Okay.
So, and the project was on this allele, this gene found only in wild mice. She was studying, the person I was working with, Sarah Lennington, was studying what that gene did with regard to reproductive success. But it meant that I had to go out and trap wild mice. So I was going to farms and stuff in the area and asking them if they had mice on their property and could I set traps and trapping mice and bringing them back to the laboratory. You could never do that now because there's so many rules about what you can bring into the animal facilities now because they're so concerned about diseases.
Oh, you brought them in?
We brought them right into the animal facility. No protections for us either. They could have carried diseases, who knows what. But it was just a very different time. So I had to trap mice, but I was competing with several others in the lab for access to this rather limited resource. And it just wasn't working out very well for me. And so I went to the behavioral genetics meeting that spring, and I was talking to some scientists that I had met over the years because Bruce had been very good about introducing me to some of these people. And that's another lesson, that networking is so critical. And so they knew me well enough to know what my skills were. And I had got 2 offers on the spot for a postdoc because I was complaining about the one that I had at Rutgers and that it really wasn't working out very well for me. And so then I had to make a decision. Now, unlike now where people go travel to see the postdoc lab, talk to the person who they're going to work with, see everything. That didn't necessarily happen back then. You know, you got a postdoc offer and you went, or you chose not to, but you didn't necessarily go and see what you were getting yourself into. And I had never been to Portland, and I didn't go see it. But I decided that I wanted to work with John Crabb, because over the years, having talked to him and interacting with him at posters and things, I could just tell that he was a very good mentor. And in addition, the research he was doing was such a good fit for the training I had received while I was in graduate school, because it was addiction research. And so I did choose to go to Oregon and go to OHSU. And I've been there ever since. I've never left there. Um, I I've been there almost 37 years, rising through the ranks, and now becoming chair of the department. And I have found John to be exactly the mentor I thought he was going to be. And the other thing that was good, and the other reason I chose him, was because he told me that there was the chance that I could stay there and develop my own research program, because the VA Medical Center was just being rebuilt, a brand new building. And he was going to hire me as a VA scientist in addition to my role as an instructor initially at OHSU. And so I could be in both programs and potentially end up in very new laboratory space in the VA. And that is what happened. So I have two jobs.
Yeah.
Yeah.
I haven't asked anybody this before, but your path makes me think of it. Do you still have mentors?
Yes, I would say I still have mentors. John, especially, I still meet with him, even though he's retired and emeritus. I lean on him still because I'm director of the Portland Alcohol Research Center, and there are always difficult decisions because people are retiring and you have to decide who might be able to replace them. And so his opinion is very important to me, and his opinion about when I should stop doing some of these things and feel comfortable handing them over. So I would say definitely John has remained a mentor. Not too many others, but John for sure.
Yeah, and I see that among my colleagues. I have my own mentors now. So,
Mm-hmm. Yeah.
um, have you experienced any eureka moments? Is what I've been calling them. They don't have to be super dramatic,
Mm-hmm.
but special moments in your research, and what led to those moments.
Mm-hmm. So I'm going to tell you, I guess I'll start with a professional eureka moment. And that would be when I came to realize that I had made the absolutely right decision to move to Portland sight unseen. I honestly didn't really like living there for a couple of years. I had gotten used to the East Coast way of life, and it was very different. But I liked my job the minute I walked in the door. And as a mentor, John was everything I expected him to be. The other thing is that there were other great mentors there, and there were men who supported women and made sure that they had what they needed to move up in the ranks. And because I had suffered some harassment in other positions, this was really good for me and extremely helpful. Um, there are a number of women chairs at OHSU. I'm not the only one, which is great. There are a number of executive vice presidents who are women. You know, I look forward to the day when the president is a woman. But that type of support has been really important to me at OHSU. And OHSU has also made strides in making sure that there is equity at the level of ethnic diversity, in addition to gender. So OHSU to me has been a great home, and that's one of my eureka things. Research. With regard to research, I think there are two. One was really early on. I was at a behavioral genetics meeting in, I think it was 1989. So that was 2 years after I'd gotten my degree. And John and I met with Robert Plomin and Jerry McLaren. And we were talking about recombinant inbred mouse strains and how recombinant inbred strains were used in plant biology for mapping genes. And could we possibly use recombinant mouse strains in that same way? And so we had a large database of behavioral data from these recombinant strains. And we also had data on genetic polymorphisms when they were doing it with restriction fragment length polymorphisms, not the way we do genotyping now. But it meant that you could take all of that information and just put it together, and you could map where certain traits had a genetic signal throughout the genome in the way we do now with really specific genotyping. So a little bit more rudimentary way. And so we decided to try it. And so we took several of the behavioral phenotypes that we had for amphetamine and alcohol and morphine, and aligned it with these polymorphisms in the RIs, and we're able to map genes to specific locations, which was a big breakthrough because it meant that behavioral researchers could use this approach without ever having to do any molecular biology, right? Which is big. You know, if you don't know how to do genotyping, right? That's huge.
Yeah.
So that kind of started the whole QTL mapping in mouse. And we published our first paper, it was 1990 or 1991, something like that. And RI strains are still being used in that way, but we have a lot more genotyping information about them, so we can more finely map than we used to be able to. I was a true believer that it worked when I collected some sucrose drinking data and did the mapping, and it mapped right to the sucrose locus that was already known in the mouse. So then for sure I was completely convinced that this was the right approach. So that was a real eureka moment to me, finding that sucrose locus. The second was quite a bit more recent. I have used so many different types of selected lines in my research. It's just a procedure that I fell in love with from the beginning, and it's because you can get a lot of genetic information using those animals. And so I selectively bred some animals for two-choice methamphetamine drinking, the way we do it for alcohol drinking. They have a bottle of water and a bottle of methamphetamine. And so I was able to easily breed animals for high and low meth intake. And I identified a QTL, quantitative trait locus, at a particular location that had a huge signal. And it turned out to be a single major gene effect that accounted for 60% of the genetic variants, which is really unheard of in the addiction field.
That is huge.
Yes, it is huge. And so it was pretty easy to figure out what the gene was. It turns out to be the trace amine associated receptor 1. It's a receptor that sits inside the cell, maybe on the endoplasmic reticulum or a mitochondrial membrane. So it's a GPCR, the types that are usually on the external membrane, but it's inside the cell, which is really interesting.
Yeah.
And it just so happens that methamphetamine is an agonist at that receptor. So it made all kinds of sense that that might be an important gene. Long story short, the reason this is a eureka moment is because when we really started to study it and try to figure out what the polymorphism is, we discovered that at the Jackson laboratory, one of the two mouse strains that I decided to use for this project, a spontaneous mutation had arisen at that location. And this is what fell into my lap. I never would have found this. No one would ever have found this if I hadn't used that particular mouse at that point in time. Because we mapped back and we found out it arose around 2005. So if I'd done this project before that, I wouldn't have found it. So it's just sometimes science is luck.
Yes. Often, I think.
Oftentimes, yeah. And so we now know exactly what the polymorphism is. And we know that it is that particular polymorphism is responsible for making that receptor non-functional. And when the receptor is non-functional, animals don't experience the aversive effects of methamphetamine.
I love that story.
Very straightforward and easy story. Yeah, so that's definitely a eureka moment.
So you've talked a little bit about this, but the alcohol field, it's very interactive, really networked. The RSA meeting is a good way to perceive that. Do you want to talk about any other people that you've interacted with that have had an important impact on your work and your career?
I think there are a few more who deserve mention. So, the first would be Bruce introducing me to John, which of course set me down this path. I want to say that John always knew to challenge me, because he called out my imposter syndrome, for example, when I came and said, John, I think I should just be your lab director because, I don't know if I have what it takes to be the PI of a lab. And he said, I'm not hiring you as my lab director. You can do this. And he pushed me to do it. So I think people like that are extremely important, especially for women in the field. He also nominated me for awards. He made sure that he suggested that I give talks when he couldn't give them. These are the kinds of things that good mentors do. They make sure that their trainees are getting what they need to succeed and to be known. You have to be known, and that's all. That networking is really important. John was also a pioneer when it came to thinking about consilience between animal models and human research. He thought about that a lot. He wrote papers about that. He met with clinicians. So he didn't stay in his silo, his, preclinical research silo, which I think is something that's really very remarkable. He did one of the most important seminal works that created quite a stir at RSA one year, that I think has been done. He got together with Doug Walston and Bruce Dudek, also, addiction researchers, and they decided to demonstrate how reliable research is using these standard inbred strains that are created at the Jackson lab. And the way they would do this is they would collect exactly the same phenotypic data at each of their labs, trying to control for everything they could. The food the animals were eating, exactly the way they were being treated, how much travel the animals went through if they were being shipped, things like that. I mean, I think they even drove them around in vans in one location because the distance wasn't the same. Mm-hmm. Unfortunately, or fortunately, many of the phenotypes were not very reliable. And this is what created the stir at RSA, because everyone had always been saying these traits are so reliable in these strains. However, if they were highly heritable, if there was a strong genetic component, the reliability was quite good. It was when there wasn't a strong genetic component. And so you can imagine that environmental factors would therefore have a greater impact. And that was really the message of the paper, but it's not the one that was heard at first. So it took time for people to sort of accept, that it was an important message and it was an okay message and not to be quite so upset about it. The next person I'd mention is Chris Cunningham. He was a professor also at OHSU. And he served as the chair of the Behavioral Neuroscience department for a while I was there. And he appointed me as vice chair with him for several years. So he placed me into leadership roles so that I would get experience in those roles in case I wanted to be in leadership later on. And that has become a big deal now that I've ended up as chair of the department, which I really wasn't planning to be, but which has happened now. So he is, his brilliance in Pavlovian conditioning and processes is phenomenal. And he's trained a number of really impressive scientists, several who are here giving great talks and have wonderful positions of their own. So Chris has been a big mentor too. I'll also mention Dr. Karl Mann. He's of Heidelberg University in Mannheim, Germany. I first met Karl when John Crabb sent me to a meeting in Germany in his place because he couldn't give the talk that he was asked to give. And I was only, maybe I'd been in Portland for 3 or 4 years by this time. I somehow ended up being interviewed on German radio with Karl while I was there at this meeting. I was giving the animal perspective of what was happening in alcohol research, and he was doing the clinical side because he's a clinical researcher. And I was speaking in English and they were translating to German. So that was quite an experience. But meeting Karl and having that experience, again, this networking, he ended up much later advocating for me to become the first woman president of the International Society on Biomedical Research on Alcoholism. So I was the first woman president of ISBRA. And now there has been another after me. So this is a big deal because it wasn't that long ago, maybe 4 years ago. Up until then, they'd never had a woman president. And so I'm happy to have seen that change. And then I'd also like to mention Carl's therapeutic research, along with John Littleton's. Both of them had a big role in getting acamprosate approved as a treatment for alcohol use disorder, which is helpful for some people. And in addition, John Littleton has done recent work looking at plant extracts, seeing some of those could be developed as treatments, and has started a company that is looking at some of that. And I'm hoping that that will be carried forward by others. So those are all people who really have had, I think, a major impact on me and the field, not to mention Ed Riley and Ann Streisguth and Carrie Randall, who did seminal work in fetal alcohol syndrome that has been carried forward, as you know, at this meeting by a lot of people. Those are all kind of people to me who have been really impactful.
Mm-hmm. What do you see as historic moments in understanding what drives substance use, alcohol in particular, but substance use in general, in some cases protects in others, why some of us are vulnerable to alcohol misuse and others not so much?
Mm-hmm. Mm-hmm. This is a favorite topic of mine. It's what my main interest is in genetics and risk for use, and not just alcohol, but other drugs. Obviously, you heard about my meth research a minute ago, and there are so many people I could mention, but I think I'll list some of the more historic people who I was lucky to get to know. The first I would say I would talk about is Mark Sheckett. He did a lot of work on family history positive and negative individuals with that family history as a predictor of later alcohol use disorder. And he also showed that sensitivity to the sedative or depressant effects of alcohol was a strong predictor of reduced risk for developing an alcohol use disorder, which makes sense, right? If you drink a couple glasses of wine and just makes you sleepy, then you're not going to be able to drink a whole lot. And so that was a kind of a seminal finding. The next I would mention is T.K. Lee. He developed a rat model with very high genetic risk for alcohol drinking. There have been a lot of selected lines produced, and his was a selected line, but he focused really specifically on other characteristics in the animals to show that they were a good model for some of those characteristics that are found in human alcoholics. So not just the drinking trait, but compulsivity and other aspects, which I think was really important. Henry Begleiter had seminal work looking at brainwave patterns and how certain patterns of brainwaves were predictive of risk for alcohol use, which I think was a really important contribution. I'll mention Harriet DeWitt. Harriet DeWitt was studying individual differences in subjective responses to alcohol in humans. So how stimulated people were or how depressed they were by drugs, not just alcohol, and showing that individual differences in sensitivity were important for whether or not they had high or low risk. So people with greater sensitivity to rewarding effects, you might predict would have somewhat greater risk. I think those studies have kind of reemerged now. I saw more at this meeting where people are starting to look at these individual differences and pay attention to them, because there are family positive individuals, family history positive individuals who don't necessarily go on to develop an alcohol use disorder or a drug use disorder. And the question becomes, why are some resistant, even though they have a strong genetic loading for that? Are there factors in the other part of their genetic background that are helping them to not become, have an alcohol use disorder? If we could figure out what those are, that might be a therapeutic avenue. So I think that that's an interesting idea. And then I guess the last person I really wanted to mention is Kathy Grant. She works with non-human primates and has done some unprecedented work in the field. She has discovered very clearly that there are some monkeys who avoid alcohol and some monkeys who drink very high levels, some who binge drink, some who drink extremely chronically. And she's been able to do biopsies, brain biopsies in these animals prior to drinking to look at the transcriptome and then look to see if there's anything that predicts their high drinking. And then after drinking, she's been able to take yet another tissue sample from these animals and look at the change in the transcriptome. This is really phenomenal work. And she's also been able to relate it to brain scans, so MRI work, as well as cognitive differences.
Sorry.
It's okay.
Okay, I'll edit that out.
Yeah.
Could you please start with that she is also related to brain scans.
Yeah. Yeah, so in addition to that work, she's also done some MRI work looking at circuitry in these animals before and after drinking. And she's also looked at cognitive flexibility and has found that those animals who have less cognitive flexibility are the ones who have greater risk for drinking high levels of alcohol, and that high levels of alcohol drinking lead to difficulties with cognitive flexibility. So I would name her as well.
Yeah. For researchers early in their careers, could be at the start or just generally early, do you have any advice to offer, and maybe including for basic scientists who want to pursue treatment development?
My advice to new researchers is really to broaden your education beyond your own specific area of research, to really be nimble. I think that if you're a basic researcher, networking with clinical researchers is important, and vice versa. You know, we have a lot to learn from talking to each other, and I think that we don't do that as much as we should. When I come to RSA, I try every time to go to at least 2 clinical symposia sessions that I would not necessarily go to, and I always learn a lot from doing that. Find it really interesting. I also think that new researchers should be nurturing and honoring their collaborations. It is what allows you to do the most impactful research, really collaborating with others, and that you should try to identify lifelong mentors who you hang on to and whose advice you really value, and to find more than one mentor while you're in school or after you're in your first job. Really important to have those people to provide your advice. And then the last thing I would say is that they need to find ways to give back, what's been given to them. I have trained a number of graduate students and postdocs, and I am just so happy to see several of them who have become professors and who have students, and to hear them say it at meetings sometimes, this is Tamara's granddaughter, because of this generational uh, tracking along, and passing along what we've each learned from each other. So that's the kind of advice I would give to them. I think that collaborations happen more easily than they used to, and we really should take advantage of that for sure. Yeah.
So through these conversations, there's been a pretty consistent theme of networking and generosity.
Mm-hmm.
It's been really warming for me to see this.
Mm-hmm.
For members of the public who may watch this video, is there anything you'd like to communicate directly to them?
Yeah. I would say that there have been technological advances in the study of genetics and of brain circuitry and of brain mechanisms that have had a major impact on what we know about the risk for developing an alcohol use disorder. And that we also know that beginning to drink alcohol before adulthood is one of the strongest risk factors there is for later problems with alcohol use, and that there's no safe amount of alcohol while pregnant. You should not drink at all while pregnant. I think the public knows some of these things, but we really need to take it more seriously than we are. It's very disturbing to see how quickly and how much the rise of alcohol use has been happening in women and in the older population. And if we don't start paying attention to some of these things and curb the use of alcohol, which has toxic effects on every part of the body and is more adverse for our health than any other drug, then we're really going to continue to have major problems in our society in dealing with this. And George Koob has done a really good job of presenting this information. And this is such a far-reaching problem for families and for individuals and for society. I think that we're getting better at identifying some therapeutics, counseling and pharmacological combinations, things like that, and that we have to continue to work together as basic scientists and clinicians to identify better ways of at least reducing alcohol use, so that the consequences are not as severe as they are for those who really use excessively.